Competitive cells are those who are capable of introducing exogenous genetic material into their system. Competition is an special physiological condition that some bacteria present naturally and can be induced in others, making them especially permeable. The permeability is due to alterations in the cell membrane. Although there are different mechanisms by which bacteria can acquire exogenous genetic information, this report is going to explain the differences between two of them: transformation and transfection. Genetic transformation is the process in which the cell captures free DNA molecules and keeps them in its interior in form of extrachromosomal replicon, or incorporates them in its genome through homologous recombination. In bacteria, the transformation is a natural process that occurs frequently. Transfection consists in the introduction of some kind of external genetic material in eukaryotic cells through plasmids, viral vectors, also called transduction, or other tools for the transference. In order to choose the best transformation method, there has to be some considerations. For example, the salt concentration in the plasmid, the membrane nature of the cell, the amount of cells to be transformed, the heat resistance of the cell, and if the cells are competent or not. Some methods used in the bacterial transformation are artificial transformation. Consists in add calcium chloride because the calcium facilitates the uptake of DNA on the cell surface, and the putro-thermal shock that allows the entry of DNA absorbed to the cell cytoplasm. Microinjection. A small amount of DNA is injected into an embryo pronucleo. This technique is commonly used in plant transformation. Transfer tramposons. The horizontal transfer genes is the transfer of genetic material between cells and genomes. They belong to unrelated species by the processes other than the production. Take 0.3 milliliters of E. coli and place it into 1.5 milliliters microtube with 30 milliliters of LB medium. Incubate for 3 hours at 37 degrees Celsius until the optic density reach an absorbance between 0.5 and 0.6 at 600 nanometers. After that time, the culture is separated into one half for the electroporation and the other half for the chemioporation. Then centrifugate both tubes 5 minutes at 5000 rpm. After the centrifugation, the supernatant is removed and the pellet is resuspended in 10 milliliters of cool glycerol at 10% and incubated on ice for 10 minutes. Then centrifugate for 10 minutes at 4000 rpm. Remove the supernatant and resuspend the pellet with 2 milliliters of glycerol at 10% and then prepare liquids, each one with 200 microliters of solution, and store them at minus 70 degrees. Withdraw a liquids and place on ice to thaw. Then 40 microliters of the electrocompetent cells and 5 microliters of the plasmid are placed into 1.5 milliliters microtube. The mix is subsequently put into an electroporation cell and then placed in the electroporator and give the electric pulse. Afterwards, transfer the mix into a microtube and then in the same microtube collocate 300 microliters of SOC medium and incubate for 30 minutes at 37 degrees with the stirring at 250 rpm. Spatulate one volume of the culture into one petri dish with LB medium and ampicillin and incubate 20 hours at 37 degrees. Finally, the colonies that grew are counted to calculate the transformation efficiency. After the centrifugation, the supernatant is removed and the pellet is resuspended in 10 milliliters of calcium chloride and incubated on ice for 10 minutes, then centrifugate for another 10 minutes at 4000 rpm. Remove the supernatant and resuspend the pellet with 2 milliliters of calcium chlorate. Add 0.1 molar and 50% of glycerol. Incubate on the ice 10 minutes, then prepare a liquids, each one with 200 microliters of solution, and storage them at minus 70 degrees. Withdrawing a liquid and place on ice to thaw, then 5 microliters of plasmid is added into each tube and place it on the ice for 10 minutes. After the 5 minutes, the tube with the plasmid is placed on water at 42 degrees for 45 seconds and then is placed on ice for 5 minutes. After the thermal shock, 300 microliters of SOC medium is added and incubated at 37 degrees for 30 minutes with the stirring at 250 rpm. Spatulate one volume of the culture into the petri dish with LB medium and ampicillin and incubate 20 hours at 37 degrees. Finally, the colonies that grew are counted to calculate the transformation efficiency.
One of the most important differences of the methodology to create chemiocompetence and electrocompetence cells is the amount of salt in the solution, because if there is a lot in the electroporator, this will be cause errors, but not in the chemiocompetence cells. The efficiency of transformation is now according to the amount of bacteria that grew in the Petri box with antibiotic. If bacteria resist the antibiotic, in this case ceonin, the bacteria was transformed correctly, but as a result of the practice, no bacteria growth was detected. One assumption is that the plasmid wasn't introduced properly in our bacteria. For this reason, the microorganisms don't have the resistant gene of ceonin, but no growth was detected with chemiocompetence and electrocompetence cells. It could be assumed that the concentration of the antibiotic was except viewed. The efficiency of commercial cells is usually higher because their processes are performed with more specialized equipment, besides that the competent cells are concentrated. For example, according to Thermo Fisher, Meganex DH10B are the highest efficiency electrocompetent cells available. They are ideal for high demanding cloning and in library construction applications.
In the case of the electrocompetence cells, the process is carried out by means of a consisting of electroporator applying an electric output during a short time interval that alters the conductivity and permeability of the cellular membrane. It's a unique physical process that requires a cellular suspension of high resistance and low conductivity to assure the insertion of the DNA and that doesn't die to bacteria. In the chemicompetent cells, the DNA by means of a thermal shock is inserted that causes the pore appearance with the eye of the calcium in the membranes that will let pass to the plasmids. This is because the calcium is united by means of electrostatic interactions with the DNA, and do that has affinity by membrane lipids.